Hello, I'm Grace Minton and this is Hannah England, and together we are genetics experts.
As of 2025, common dental practices included dental fillings, veneers, and dentures as a way to resolve dental loss and tooth decay. As far as prevention strategies go, there are none that are 100% effective. People can maintain a healthy low-sugar diet, brush and floss twice a day, and return to the dentist twice a year for their annual visits. However, there is no guarantee they won't be affected by some sort of oral disease at any point in their lifetime.
As the science behind genetics advances, we can further improve different parts of medicine, including oral and dental health. Today we're going to talk about how CRISPR, a gene editing technology, can change the dental industry.
CRISPR stands for Clustered Regularly Interspaced Short Palindromic Repeats. This specialized portion of DNA consists of three key characteristics that are important to take note of: the presence of nucleotide repeats, spacers, and PAM sequences.
These components are vital to the function of CRISPR, as they each play an active role in the gene editing process. First, the nucleotide repeats and spacers form the genetic framework that allow CRISPR to identify and target specific DNA sequences, mimicking how bacteria recognize viral DNA.
During gene editing, Cas9 enzyme, guided by RNA molecules, both cRNA and trace RNA, derived from these spacer sequences, locate the matching DNA in a target organism and make a precise double-stranded cut at sites marked by the PAM sequences, allowing scientists to add, remove, or alter genetic material with high accuracy. Now, in order to use CRISPR and Cas9 as gene editing tools, repair mechanisms must kick in to fuse the two ends together. These two ends are fixed by inserting a sequence of nucleotides in the gap. This sequence can be any nucleotide sequence of a scientist's choosing. In this case, we will be looking at a sequence that would make dental pulp stem cells to be specific, also known as DPSCs, produce more of a beneficial protein known as BDNF, or brain-derived neutrophobic factor.
In order for scientific development such as CRISPR to go into effect, repeated trials of experiments must be run and deemed successful. Therefore, an experiment using CRISPR was performed by the University of Illinois Chicago, New York University, Langone Medical Center, and University of California, San Francisco in July 2025 on mice, aiming to generate dentin, the hard, dense tissue that forms the main structure of a tooth, lying beneath the enamel, which is the outer layer, and above the pulp.
Scientists first collected dental pulp stem cells, or again known as DSPCs, from human teeth. Using the CRISPR gene editing, they changed the stem cells to produce more of that helpful protein called BDNF, which supports growth, survival, and function. They then confirmed this change in the lab.
Next, they drilled a small hole into the tooth of a mouse to create a tooth injury. This simulated tooth injury or tooth decay that is often seen in humans. The edited stem cells were then placed into the injured area along with supportive materials. The tooth was sealed to protect it, and after some weeks, the researchers checked to see how well the tooth healed.
The researchers found that the treated teeth grew back with more healthy dentin. This means that the insertion of CRISPR-edited dental stem cells could potentially replace traditional dental fillings.
Instead of using synthetic filling materials to fill cavities or repair damaged teeth, the edited stem cells are transplanted into the injured area to encourage the tooth to heal itself by growing new dentin naturally. This biological approach stimulates the body's own repair mechanisms, promoting regeneration of the tooth structure rather than just filling the hole with an artificial material.
These applications leverage CRISPR technology's ability to precisely edit genes in dental stem cells or oral bacteria to promote tissue regeneration, fight infection, and correct genetic causes of dental diseases. This broad potential could transform dental treatments by enabling personalized biological repair and preventative strategies.
While there are many positives that come with CRISPR, there are also many downsides. There's also lots of controversy surrounding gene editing technology.
One prominent ethical drawback of gene editing within the field of dentistry is as follows. CRISPR-based dental treatments are expensive and would likely be inaccessible to many, widening the health disparities in dental care, especially in underserved populations.
Additionally, CRISPR-based dental treatments pose the risk that gene editing will damage the genome unintentionally by either cutting or altering DNA in the wrong places. This error is more commonly known as genome vandalism and can lead to unintended mutations, cell malfunction, or death, and even a chance of tumor formation.
CRISPR, as a gene editing technology, can help improve dental regenerative medicine in so many ways. CRISPR allows for very accurate editing of specific genes and is ultimately faster and more flexible than older techniques. In our case, these older techniques would be DPSCs, again, dental pulp stem cells.
CRISPR also holds potential in correcting genetic mutations that cause inherited diseases. In dental medicine, this could mean correcting genetic defects causing enamel disorders. As CRISPR continues to help us develop gene editing techniques, we can regenerate smiles using the power of science.
Thank you.